Let's see if that works. Excellent connection. Okay. Okay, awesome. So we're live streaming. That's good. Let me introduce you before we get started. Let's see. Oh, yeah, we've got a quorum. I think we can. Proceed. As usual, we always appreciate when people turn on their videos and get more involved. As long as that's okay with you, Mor, like people can interrupt and ask questions.
Yeah, of course.
Okay, awesome. So we're honored today to have Mor Nitzan, who teach us, and she's a senior lecturer and assistant professor at the School of Computer Science and Engineering at Hebrew U in Jerusalem. Also affiliated with the Raka Institute of Physics and the Faculty of Medicine. So her work spans lots of different fields. She got a PhD in physics and computational biology at Hebrew U with Professor Hannah Margalit and Professor Ofer Biham. During that time, she did a lot of transcription regulation and had a lot of impactful papers on long-range network effects of small and micro RNAs. I was going through the Google Scholar, and there were medical papers on amnesia and the microbiome and bacterial switches. So there are all sorts of cool other stuff. When she, during her postdocs, she postdoced at the Broad Institute in Harvard, with Professor Nir Friedman and Aviv Regev in conjunction with Hebrew U. And there she did some amazing stuff, which I think we're going to hear about a little bit today, with reconstructing the spatial position from single cell transcriptomics, as well as still publishing in other fields like neuroscience and economics. So I thought that was cool. And also was a joint John Harvard and James S. McDonald Fellow at Harvard, working on random matrix theory with Michael Brenner and optimal testing for COVID at some point. So a lot of cool stuff, a lot of diverse profile, and we are very excited to hear from you today. And thank you so much for being here.
Yeah, so thanks a lot for the invitation. I really like the concept of this seminar series. So I'm looking forward to both parts, both the more usual type of talk and discussion, the first one, and then we'll have like an informal discussion later, I guess. Right, so today I'll talk about inferring collective spatiotemporal organization in biological systems from single cell data, and feel free to ask questions and interact along the way. Okay. So in our group, we're interested in organization principles that bridge biological systems at different scales. And I guess this echoes with many of the interests of the people here. Basically, how can we think of interacting systems starting from gene regulatory networks where different genes excite and inhibit each other's function, but then each one of these networks and the collective behavior that it manifests actually lies within a single cell, and these cells interact in turn with each other to lead to the collective behavior that we see in tissues that also lead to their collective function or dysfunction at times. Right, and we're interested in many different questions and concepts that bridge these different scales, starting from information encoding and self-organization from cells to tissues. I guess can we maybe mute one of the participants? Thank you. Okay, and then also thinking about decision making on a cellular level and how that manifests, starting from the internal gene regulation networks within and between cells, all the way to decision making both on the individual cell level and collective cellular level, and division of labor between cells and tissues and whole organisms, right? And we're thinking a lot and working on these different directions using different methods from physics and computer science. But today I'm actually going to talk mostly about something that underlies many of these challenges, and this is how can we meaningfully represent cells or cell populations in terms of their expression or phenotypes, and how that and how can we learn it from data to get a better understanding of how cells are organized in time and in space, to then be able to better understand these higher level questions, I would say, about information encoding, self-organization, and so forth. Right. So to do that in connection with real data and real systems, we need some sort of data, and luckily we're in the midst of a revolution in experimental biology that has to do with our ability to measure cells at individual levels, or the more specifically the gene expression of cells or other characteristics of whole cellular populations. So in general, the picture that we should have in mind is that we start with a tissue. That tissue in general needs to be dissociated into single cells, and then we can measure them or sequence them using, for example, single cell RNA sequencing. And then we get this kind of matrix representation of genes by cells that tells us for each individual cells, for each individual cell out of the, say, tens of thousands or hundreds of thousands in our sample, what is the activity level of each one of its genes, right? So that basically means that for each cell, we now have a vector of about 20,000 entries, depends on the experiment, that is a proxy for this cell's identity or behavior. So that's great, but this data is very challenging for many different reasons. One of them, and one of the more interesting ones, I guess, is that spatial and temporal contexts are lost. And what that means is that as I was describing the very high level workflow of how do we get such data, what I told you is that the tissues have to be dissociated, right? So cells basically lose their original spatial context, and we no longer have information about where they were in the tissue and which other cells they communicated with, or how gene expression was generally patterned across the tissue, which can be crucial if we want to learn something about many different biological processes. For example, the spatial body plan of during development, right? So that's about space. And then in addition to that, the measurement itself, to measure the cell, we need to kill it, basically. So we only get one measurement per cell, right? So we can't follow explicitly any dynamical processes that the cells are going through, which is something that obviously is of main interest to us. So both spatial and temporal contexts are lost. And then in addition to that, the data is partial. So we don't actually have access to all the cells in the tissue, and we don't have access to all the genes, and the data is noisy and it's sparse. So we need to deal with these different aspects. And that's why over the past decade, this push in technological advancements was accompanied by many different computational methods and advances on many different fronts of how to deal with this data and how to learn from it about biological behavior. So that journey as a community started mostly with trying to reveal heterogeneity of cellular populations and trying to understand which cell types are in this data now that we have access to individual cells and not just an average expression over, say, millions of genes in a sample. And then from that, over the past few years, there have been an increasing number of works that have to do with inferring hidden biological structures and processes, such as temporal trajectories, spatial structure of the underlying tissue, interaction networks, both within and between cells, and differentiation structures. And we've been interested in these different directions over the past few years and thinking a lot about how can we push these frontiers forward. And I'll start by telling you briefly about how this journey started from inferring spatial context of cells, right? So in that setup, what we should have in mind is that we have our input, we have our single cell gene expression profiles, right? So this huge matrix of genes by cells that we get out of, say, single cell RNA sequencing experiments. And as I said, the spatial context is lost, and what we want to computationally infer is a virtual tissue, right? We want to infer back how the different cells were organized in the tissue of origin from which they were dissociated, and then also how different genes or different features in this matrix are expressed across the tissue, right? And again, if we're thinking about development, a developmental context, that would mean basically mapping the body plan of different organisms. Okay, so how should we think of this challenge, right? And up to a few years ago, the usual way that people thought about it is that we had to have additional information in the form of a reference atlas. For example, an imaging data set for a small subset of genes that tells us how these genes are expressed across the tissue, usually at lower spatial resolution than in single cell data, right? So here, for example, we have this kind of maps for 3 genes, right? Gene 4, 25, and 173. And then we can look back at our original single cell matrix. We can look only at these 3 rows, right? And then ask for each individual cell how well it matches each of the positions in the tissue according to the reference atlas. So that has several drawbacks, this kind of approach. The first one is that many times the reference atlases that we have are insufficient quality or don't include enough genes in them or lower resolution than we'd like. And then also for many different tissues, we actually don't have such reference atlases, right? So we can't really rely on this for the spatial reconstruction task. So what can we do without such a reference atlas? And then in that case, a very useful analogy that we can have in mind is that we're faced with the challenge of solving a puzzle, right? Where we're trying to take the individual puzzle pieces or single cells and try to find the optimal configuration in physical space that gives us a nice picture at the end for a puzzle or a tissue that is consistent with what we know from prior knowledge and prior experiments. And okay, so for puzzles and pictures, it's very intuitive, right? What it means to have a nice picture at the end, and we can talk more about that and how it, how the analogy actually extends to many different directions. But I'll skip that and just say that for when we're thinking about organizing cells to get a tissue that makes sense at the end, we need to have an assumption about, if I now give you, say, 10 different potential organizations of cells in space, you need to be able to tell me somehow which ones of these organizations are more consistent with what we know about tissue organization either in health or disease, right? And we started with the simplest assumption that we could come up with, which is a structural correspondence between expression and physical space. And what it basically means is that at least for simple tissues, we assume that there's this kind of smoothness, overall average smoothness in gene expression over the physical tissue. Or another way to think about it is that we would expect cells that are close to each other in the tissue of origin to have, on average, relatively similar expression profiles relative to some other two other random cells in tissue that are farther apart. And this assumption actually works very well.
Question?
Yeah, sure.
And maybe you just have to tell us more about the system. I'm just wondering, like, the way the system is prepared for single cell RNA-seq, does that like impact the way the tissue is grown at all? Or
so can you repeat so can you repeat the question?
I'm just wondering if the way the system is, like, I assume that it has to be grown somewhat ex vivo in order to be.
So not necessarily, right? Yeah. It depends. Whenever you can get, say, a biopsy, right, you can take it in vivo.
You can put it right into the single cell.
Yeah, so it doesn't have to be in a synthetic setting.
So you've preserved all the spatial information.
Right. Okay. So assuming here that you're preserving the spatial information in natural systems, and now you're actually breaking them apart like in a puzzle, and now you're trying to piece them back together.
Okay.
That's okay.
Cool. Thank you. Yeah.
Sure. Okay. So what we found is that for many different systems, both in early development and for simple systems that are mostly whose expression is mostly driven by external gradients, say of oxygen, morphogens, then this structural correspondence assumption holds very well. And this is what we can see here for, as an example, for the Drosophila embryo, the fly embryo at early stages of development, where we see on the y-axis the pairwise distance in expression between different cells and how it monotonically increases with, on the x-axis, the pairwise distance between cells in physical space, which reflects this kind of smoothness assumption in gene expression space, right? So now that we have this assumption, that would mean that given 10 different configurations, I would be able to tell you which one is most consistent with this assumption. But of course, it's a hard computational problem. We can't actually go through all different spatial configurations. So we need an efficient way to do that. And the way we did that in the past is through a computational framework that we called NovaSpark for de novo spatial reconstruction. And the basic idea is that if we're thinking of the original population of cells in gene expression space on top, right? So it's a 20,000, say, dimensional space. And we'd like to map these cells to the tissue of origin. So you can see the different potential locations in the target tissue on the bottom. And that can be a 3-dimensional space, or maybe a 2 or a 1-dimensional space if we have some inherent symmetries in the system. And what we'd like to do is map cells in the way that captures the structural correspondence between pairwise distances in these two spaces, right? So an additional complexity is that the cells in the high dimensional expression space tend to lie in low dimensional manifolds that capture the simplicity of biological processes that these cells are encoding. So overall, we'd like to measure pairwise distances maybe over this manifold or over local graphs, right? And then we can get these two distance matrices for expression in physical space. And we'd like to match basically cells to locations in a way that preserves these distances, right? And we chose to do that by probabilistic optimal matching, which I'll say a few words about in the next slide. And then also, optionally, I told you that we don't want to rely on a reference atlas, but if we do have access to a partial reference atlas, we'd like to be able to use it, right, to enhance our reconstruction. And then if we can do all that, we'd be able to reconstruct the original tissue of origin or get a representation for a virtual tissue. Okay, so this flow can be translated into an optimization problem where we're trying to find the optimal coupling or a matrix T of cells in expression space by locations in physical space. And what we're trying to optimize is basically what we're trying to do is to minimize the discrepancy between distances in expression physical space that captures our smoothness assumption. And then we're interpolating that with minimizing the discrepancy between our mapping and what we already know about the system, for example, from given a partial reference atlas that we may have access to. And then in addition, we're thinking about this problem probabilistically. So we have an entropic regularization term as well, and that's subject to marginals over cells and positions, which lets us, again, use different types of prior knowledge about, say, different densities of cells in different parts of tissues or different qualities of single cells that were sequenced. And for more details, I guess I'll refer the whoever is interested to our publications. And then if there are other questions, I'd be happy to talk about it later. Okay, and I'll also say that this optimization problem falls into the mathematical framework of optimal transport, where in general, we're trying to optimally transport one distribution, in this case of single cells, into another distribution, in this case of location. And it's a beautiful mathematical theory that we're drawing a lot of inspiration from, both for these works and for ongoing works that I'll talk a bit about later on. Okay, so we have this framework, NovaSpark, that's probabilistic in a sense that each cell is assigned a probability distribution over locations and not just a single location. And that makes the problem feasible computationally and robust and makes sense in many different biological settings. It's flexible in the sense that it allows us to incorporate different types of prior knowledge, like a reference atlas, cellular densities, and so forth. And we've shown that algorithmically it's feasible for large data sets. So now that we have it, we can actually use it to try to reconstruct different types of tissues from single cell data. And I'll just show a very quick example for the Drosophila embryo from before. So here you see the expression patterns of 4 different transcription factors from experimental data, and then synthetically dissociating it, trying to piece them back together like a puzzle using NovaSpark, using only 2 marker genes or reference atlas of size 2, which is very small. We get a pretty perfect reconstruction for all the different thousands of genes that are expressed in this early developmental stage. And that also means that now that we have our virtual tissue, we can do different things like infer spatially informative genes, so genes that vary informatively over the tissue and probably are related to spatial biological processes. And as we would expect, we find that transcription factors are among the top spatial informative genes, as we know from classical genetics. Okay, and we can do that for many different systems, and the code is and tutorials are on GitHub, and whoever's interested is welcome to try it. And yeah, and if you have any questions, feel free to reach out. Okay, so that was about how can we take single cell data and reconstruct the original spatial configuration of the tissue that the cells came from, right? But we've been thinking a lot about how can we actually go beyond canonical spatial reconstruction. So both to different types of domains, right? So taking single cell data and, for example, not only reconstructing space or spatial structures, but also reconstructing other types of domains like temporal processes and additional biological processes that the cells are encoding using the same type of probabilistic ideas. so that's one big direction. And then the other is how, even if we stay within the spatial context, how can we go beyond canonical tissues in which we can rely on this kind of smoothness assumption or structural correspondence between expression and physical position, which obviously breaks for many types of more complex or disordered tissues, right? And maybe the extreme is the example of cancerous tissue, right, where we can definitely not rely on the smoothness assumption. Right. So to deal with these kind of questions, we've been developing a framework that we termed TACO, where we're using, I would say that the core is related to similar types of probabilistic reconstruction ideas, but now we're asking, how can we actually effectively and scalably integrate different types of knowledge when we can't rely on the smoothness assumption, right? So say we take high quality single cell data as before, and now also add maybe a lower resolution spatially informed data, like spatial transcriptomics or slide-seq data, where we may have information about how different genes are expressed over space, but at lower resolution than single cells, right? And in that case, this work is led by Noah Moriel, a great PhD student in our group, and also Simon Mages and Inbal Davidi. They've been pushing these works, and it was a joint effort with many different collaborators. And what we're trying to do here is basically ask, how can we probabilistically transfer annotations from high quality data to another data set where we have maybe an orthogonal piece of information that we care about, like the spatially informed data, like in the slide-seq case. And if we can do that, we can get on top of the slide-seq beads, we can get cell type annotations based on the single cell data, and then also aggregate different environments, different cells, different beads to get more large-scale microenvironments or regions that tells us something about the overall behavior of the tissue, both in health and disease. Okay, so this was all about how can we take single cell data either by itself or together with an additional type of information and uncover a single layer of information that we care about, for example, the spatial configuration of cells. But then a greater challenge, I would say, is how can we actually okay, so that's based on the realization that in fact cells simultaneously encode multiple layers of information within their gene expression, right? So we have this opportunity to ask not only how the cells are organized in space, or not only how they're organized in time along some temporal process, but actually taking this realization that the cells encode both types of information simultaneously, how can we decouple that information and recover both at the same time, right? So say take single cell data, recover the spatial organization of cells, but also ask which other processes are the cells encoding? Or more generally, how can we disentangle different layers of information, right? About cell type and temporal processes and spatial organization and interaction networks and lineage structure and all of that from single cell data. And that kind of question is challenging, but it's very promising, because if we can do that, then that would mean that from single cell data, we'd be able to simultaneously capture, for example, for the fly embryo that I talked about before, both the spatial organization and also how different lineages are formed and how different cell types are formed over development, or for liver lobules, where we know that cells vary their expression both across the lobe, across space, and also across the time of day, how can we actually discover this two-dimensional variation, right, in both space and time? And also, for example, thinking about brain development, right, we'd be able to simultaneously capture differentiation structure and spatial organization and how that forms over developmental time. So we have a lot of reasons to devote our efforts towards answering this question. And I wanted to give two examples for directions that we've been working on towards this goal. So the first one is a project led by Zoe Piran, a wonderful PhD student in our group, that developed a computational framework termed SIFT for signal filtering. And the idea there is to emphasize cellular processes by filtering gene expression of different processes through projections. And I'll say a bit what I mean by that. So if you look at this figure that I have here, we can think of this kind of simple scenario where the cells are encoding two different types of layers of information, or two different processes, process A and process B. And what we see at first is that the strong signals that we can capture from the cells is related to process A, right? So if we, for example, if we would just cluster the cells in space based on their similarities in gene expression, we would see these clusters that capture the clusters related to process A, right? So that can be, for example, cell type. But what we'd actually like to do is now say, okay, that's great, but we know there's more information in that data, right? So we'd like to filter this layer that we were able to reconstruct, to filter it out. And then by using SIFT, hopefully we'd be able to reveal this underlying information layer where now the cells are clustered according to process B, right? In this case, according to their different shapes. And the basic computational idea here is what we're trying to do is take the original data, in this case, the single cell gene expression data, X, and we're subtracting from that a projection over the original data that captures similarities between cells according to the process that we're trying to filter out, right? So what we're trying to do is get a new representation of the data that does not capture the structure that we were able to reconstruct, for example, the spatial structure that I told you before. And now we're trying to look for other similarities between cells that hopefully we'd be able to decode as another layer of information, another biological process. And I'll give a concrete example for that. So here, what we've done is take single cell data from mammalian liver lobules, right? So we have this gene expression matrix of cells by genes. We can use NovaSpark, as I've shown before, to get a spatial reconstruction and get this structure of for how gene expression changes from, in this case, from the central to portal vein in each one of these repeating units in the liver. And that tells us something about how spatial zonation is expressed in that tissue, and that's driven by gradients of blood and therefore oxygen and also morphogens and so forth. But now, as I told you, what we want to do is actually say, okay, this is nice, but we want to ask what else are the cells encoding, right? So using SIFT, we're filtering out the spatial structure that we've decoded from the data to try to recover how the cells vary their expression along the circadian rhythm, along the time of day. And what we see here is on the y-axis is how well do we do in trying to capture the expression across the time of day, so the correlation to the temporal trajectory in the y-axis. In blue, you can see how well we do with the original data, and then in orange with the filtered out data. And you can see that we do a lot better once we filter out the information about space that is not related to how the cells vary their expression over time. And on the x-axis, you see how that changes as we add more and more marker genes, or how we add an increasingly growing reference atlas to get the temporal reconstruction. Okay, so that was one direction I want to share with you that hopefully we'd be able to share formally soon. And then another direction I want to share is based on a different perspective. So I'll tell you about that a bit now, and that has to do with identifying signatures for topological universality classes in single cell data. So what we've realized at some point is that if we want to decouple different types of processes from single cell data, one of the anchors, one of the conceptual anchors we can use for that is that many of the biological processes that we care about chart distinct topological classes in gene expression space. So for example, if we're thinking about a differentiation or a developmental process on the left, then we would expect to set the cells going through this process, and if we look at them in gene expression space, to approximately chart a bifurcating tree. Or if we're thinking about different processes like the cell cycle or the circadian rhythm, in the middle, we'd expect the cells to chart a cyclic process in gene expression space. Not necessarily a perfect circle, but a cyclic process that tells us that the cells are going through certain process and getting to the starting point at the end, right? Like over and over again each day with the circadian rhythm. Or if we're thinking about a spatial structure on the right, we'd expect the cells to chart a three-dimensional grid, or maybe lower than that if we have some symmetries in the system. So we were thinking, how can we use this idea that we have these distinct topological classes? Can we get some hints about which type of class is actually represented in our data? And if we can do that, maybe we can also use it to decouple different biological processes or structures. And what we went for is thinking about a spectral analysis perspective. So, okay, so if we now want to ask what kind of signatures we might hope to recover from single cell data that are related to the different classes, we first need to understand what would we expect, what is our null hypothesis, right? So starting from independent gene expression profiles, where the expression of different genes in different cells is uncorrelated, then we're in a random matrix regime. And then in that case, we know what would we expect the eigenvalue distribution over this data, this matrix, or the covariance matrix of this data, or in this case of these single cell data, to look like, and that's a Marchenko-Pistorius distribution. So this is what it looks like when we look at the probability of eigenvalues over their value. And then we can also look at it differently, and you'll understand why I show it this way in a second. And we can look at the log-log plot of the eigenvalues versus their rank, if we now order all the eigenvalues of the covariance matrix of the original data. Right, so in that case, we get a near zero slope for high eigenvalues that diminishes at some point. So that's what happens if we have independent profiles, right? But of course, that's not what we have for real systems. In real systems, different genes and different cells are correlated, right? Different genes are correlated, the expression or the activity levels of different genes are correlated due to gene interactions, and the expression of different cells are correlated due to their organization in space or their ordering across different temporal processes and so forth, right? So we would have these different correlation structures. And now the question is, can we identify from the spectral signatures what type of biological process are the cells encoding, right? So taken as an example, a lineage model, right, where the cells start from, say we have a cellular population that starts from stem cell that goes through different changes. It divides, goes through different changes again, divides again, and so forth till we have our leaves of the tree or terminally differentiated cells, right? So now when we take samples from this tree, we can show both analytically and in simulations that the spectral signature that we get is substantially different than that of a random single cell population without any correlations. And specifically, we get parallels of large eigenvalues for this kind of tree-like structure that captures both lineage and differentiation and developmental trajectories, right? And we can see that in real single cell data. So this is an example for this kind of signature that we can detect for single cell data taken from the epidermis or skin. So we see these type of parallels of large eigenvalues that I described that are diminished when we shuffle the data and basically lose the correlations between the different cells, and that's a general phenomenon. So we can see that for different tissues and different systems where we would expect the cells to encode strong signals about differentiation or developmental processes, about tree-like processes. And when we look at the genes dominating the high modes, we see that they're enriched for this kind of hierarchical process like differentiation, developmental, and so forth. Okay, so that's good. But what we did in this work is basically say that we can detect the signature for tree-like processes in the data, right? But we want to go beyond that and ask whether we can leverage this kind of spectral approach to now also manipulate the data, right? So to either enhance different processes and filter out others, and so forth. And that's what we did. So and specifically, this is
sorry, can I interrupt quickly?
Sure.
Yeah. So you said you shuffled the data before you take the power law as a result of the shuffling, or
no, so it's not a result of the shuffling. What I just showed, it was like a sanity check that when we shuffle the data, we no longer see the power laws.
Oh, okay. You lose the power law when you shuffle.
Yeah.
But it's not, the point is that the tree structure doesn't give you Marchenko-Pasteur.
Exactly.
Gives you the power law distribution instead.
Right.
Okay. Interesting.
More, I have a follow-up question. So in constructing the analytical prediction or expectation for the hierarchical distribution of eigenvalues, how many parameters go into that? What do you have to estimate?
Right. So eventually, what we see is that the power law itself, its slope depends on the ratio between, if we're thinking about the simple model, right? It depends on the ratio between the number of changes in each branch and the overall number of genes, right? Mm-hmm. So how much do you vary your expression between each bifurcation?
I see. Okay.
Mm-hmm.
And that's something you can just sort of estimate in some handful of your genes and
yeah. So depending on the system, right? What we did in this paper is that it was actually hard to estimate it directly for different reasons, but what we showed is that it varies along the direction that we would expect. So the slope increases or decreases as, for example, we go forward or backward in developmental time, either natural or synthetic development.
Oh, cool. Okay. Thank you.
Okay, so I guess the final project that I'm going to tell you shortly about is led by two students from our group, Jonathan Kareem, a master's student who's done a great job with this, and Yoni Bohnfeld, who's an undergrad doing a project in our group this year. And it's been a great collaboration, and they're proposing this more general spectral framework called SC Prisma. And the basic idea is to use this kind of spectral template matching based on the realization that different biological processes are associated with different topological classes, right? So in this project, we focused mostly on cyclic or periodic processes, and we show that we can also extend this to linear processes. And using a series of optimization problems, basically, how can we go from the original single cell data or single cell matrix to different representations of the data that either highlight or filter out the spectral signature that we would associate with different biological classes. So again, to make this more concrete, here's an example for what we get for a population of HeLa cells that we know that should relatively strongly encode the cell cycle signal in their gene expression, right? But even so, when we look at the raw data, we look at the original PC representation, principal component representation of the original single cell data. We can't really see anything, and then another way to look at it is a polar plot that we see right below it, and looking at subsets of genes that we know are related to different phases of the cell cycle. And again, we can't see any reasonable distribution of different phases along the cycle. And that also shows, for example, for two different genes on the bottom that we know are strongly expressed in specific phases of the cell cycle, they're pretty much uniformly expressed in the raw data, right? But when using the spectral template matching and enhancing, but not overfitting to a cyclic structure, we can see the cycle emerge on the top. Right below that, we can see that the ordering of the cells along it corresponds to different phases of the cell cycle in the right order. And then also looking at specific genes on the bottom, we can see that they're expressed along these phases as we would expect, for example, in M and S phase in this case. And then another very quick example, I told you before about the single cell data in the liver lobules. So looking at it from this perspective again, here we can see the raw data of the single cells, or specifically of hepatocytes from the mammalian liver. So we see the raw data or a piece of representation of it, where the cells are colored on the top by different time points along the circadian rhythm, and on the bottom by their different locations across the spatial axis of the liver lobule. And then when we use our approach, SC Prisma, we can either enhance the cyclic process in the data, so in this case, the circadian rhythm, showing how the cells are ordered along the time of day, or filter out the cyclic process, therefore enhancing, in this case, the linear structure of the data, because we have this one-dimensional symmetry corresponding to the spatial location of the cells in the liver or in liver lobules. Okay, and hopefully you'll be able to get access to this method and manuscript in the next few days or week or so. Okay. Right. So in general, disentangling multiple structures in single cell data, what we hope it will do, that we've showed for different cases, but we have many different ideas and ongoing directions on that front, is that it would help us to identify spatiotemporal and informative genes, identify gene groups related to different signals or structures, infer goal-turn networks, driving the dynamical trajectories that we recover, and also uncover design principles related to different biological domains. So I'd just like to end by thanking our amazing group members and different collaborators and funding sources, and thank you for listening.
Thank you so much, Mor.
Thank you so much.
That was a tour de force of
Yeah.
making sense of single cell. So that was amazing. Why don't we turn to some questions about the content of the talk itself, and we'll keep recording for that.
Sure.
So I guess I'm wondering about some of the technical details here. I mean, it occurs to me that when you're trying to reconstruct space, you're mapping this 25,000 dimensional space onto 2 or 3 dimensions, which is very similar to what these dimensionality reduction techniques are after, like
right.
CC or UMAP or something.
Okay, so that's a great question. Okay, well, I guess I know what the question is. So what's the core difference? Right?
Yeah, go
yeah. So this is great, and I should probably highlight it. I should have probably highlighted it before you asked the question, but I guess there are a few core differences. So thinking, for example, about PCA or TISNE or UMAP and how these approaches are different from it. So going to the first project I described, right, of Novospo, right? Mm-hmm. So in that case, there are a few main differences. The first one is that we're thinking about this whole problem probabilistically, right? So in any one of the methods that you mentioned or other usual dimensionality reduction methods, they're always deterministic, right? So we're taking a sample in high dimensional space and we're mapping it to a point in low dimensional space, right? So 1 point to 1 point. Mm-hmm. And then that's many times problematic because we often don't have enough information to have this kind of one-to-one mapping, or it just doesn't make sense because of the context that we have, right? So if we're trying to map single cell data that's taken from a similar tissue to so if we have 2 types of measurements, one of single cell data and another of spatially informed low resolution data, right? The number of samples that we have in each dimension are different, right? So in that case, doing a one-to-one mapping doesn't really make sense. And that's why we think about it probabilistically. So each cell is assigned a probability distribution over the lower dimensional data, over the lower dimensional structure, for example, the spatial structure, that makes this feasible conceptually. And then also it has these other advantages of making the mapping more robust and computationally efficient, because many times, if you don't have enough information, if you don't know enough about say a particular group of cells, what you'd like to do is be able to reflect that in the answer that you get, right? And not get just like a random assignment of the cells, but in this case, maybe get like a more uniform assignment and then have the reflection that you just don't have enough information to get a one-to-one mapping for these particular cells. So that's one thing. And then the other thing is that in all of these methods like PCA and TISNE and UMAP, you don't have the ability of integrating other types of prior knowledge that you many times have in these kind of biological systems, right? So either the spatial expression of a small number of genes, or there are many different, or maybe you have information not only about the RNA expression levels, but also about proteins or something else, right? So you'd also like a flexible method that lets you get an embedding that's consistent with what you already know about the system. So I think these are the two main differences or advantages of these kind of approaches. And then there are other more, technical differences. But I'll also say that getting a better initial reconstruction also lets us answer the next line of questions about how can we now decouple different processes from each other, right? So not get just a single representation, but also now say that we want to get several representations simultaneously that reflect both space and time.
Yeah, I really like that idea of decoupling that you're presenting. I mean, it occurred to me that like in linear data, right, when we do PCA or something, we get that second eigenvector immediately. But if the data is highly nonlinear, then you might need to do some sort of subtraction in order to get at.
Yeah, so these are two different points that are related to each other in a way, right? The first one is that linear embedding may just not be enough because the data may lie on nonlinear low-dimensional manifolds, right?
Sure.
So you would need a nonlinear embedding. That's one. And then a second point is that the data actually may lie on multiple different manifolds simultaneously, right? Reflecting multiple different biological processes. So these are like two challenges that we need to cope with.
I see what you're saying. Yeah, and you have to disentangle them to, yeah, that's
exactly
very cool.
Yeah.
Thank you.
Mm-hmm.
We have one question in the chat from Ali. I don't know, maybe I'll just read it. Thanks for the excellent talk. 2 questions. 1, how does NovaSpark perform in less spatially organized tissues such as tumors? And 2, did you compare NovaSpark with spatial genomics methods?
Yeah, so for the first point, yeah, so I guess, as I said at some point in the talk, when we get these highly disordered tissues, like in the case of cancer, we can't rely on a smoothness assumption, right? So if we would just try to run NovaSpark with no additional data, just relying on smoothness of expression, it would not work well, right? And then in these cases, thinking of the original NovaSpark framework, then we have this ability to interpolate between the smoothness assumption and a potential reference atlas, right? So as the tissue becomes more and more complex or more and more disordered, we'd like to shift the weight towards using a reference atlas. And if we don't have any, then we would not be able to do well in that scenario. And then the other thing, as I talked about after NovaSpark, is that we're thinking of more general ways of incorporating multiple types of experimental data using TACO, for example, right? So mapping or annotating between the high quality single cell data that we may have access to and the cellular identities, and map that information onto, say, a lower resolution spatial map. So that's, I guess, the first question. The second question, I'm not sure I understand the comment. Spatial genomics meaning probably spatially informed methods, like slide-seq, maybe? Is the person?
Yeah, Ali's still here. If you want to unmute and ask, you can go ahead and do that.
I can also just assume that that's what you meant, and then say that in the original paper, so we've compared it to exactly such as slide-seq. Yeah, so in the original NovaSpark paper, one of the systems that we test or that we analyze is actually slide-seq data for cerebellum slices from mammalian brains, and we show that if we take this data and synthetically dissociate, we can reconstruct it back, like the puzzle that I talked about, and get a better and better representation of the original slide-seq data. Mm-hmm. And again, we can do better once we have like multiple types of data sets like slide-seq plus single-cell RNA sequencing.
Mor, I have a quick question.
Okay.
Hey.
Go ahead.
Thank you. Hi, Mor.
Hi.
Thanks for the great talk, by the way.
Thanks.
I was curious about the information index. So I was wondering, in the context where you're breaking down into few processes, would you be able to recover information index of a gene driving that process? So spatial information index is what you showed. That was in the spatial coupling context. So I was wondering if you can do that for the processes or.
Right. So conceptually, it should work similarly, either, when we talk about spatial processes or temporal processes, right? As long as we have, as we get a meaningful representation or reconstruction for the original process, then we'd be able to also recover what are the, say, process-dependent informative genes, right? So either spatially or temporally informative genes. There are many different ways of doing that. Then, once you have a reconstruction, how do you actually say which genes vary informatively across space? So what we've done for that is use one of the available methods, like spatial autocorrelation analysis, or there are other approaches for that. We're also thinking about how to more generally infer spatiotemporal informative genes, even at the point where you don't have a perfect reconstruction of the underlying process. But these are, we have some early thoughts about that, but we haven't done that yet.
Okay, thank you.
Mm-hmm.
Hi, Mor, excellent talk. I had another related question about the connection between UMAP and NovaSpark. So I noticed that in some tissues, for example, in lung maybe, so even if you do just UMAP, there are already some spatial structure from one axis to another, showing the direction of the differentiation, which is somehow like correlated with the spatial organization.
Right. So okay. So the point is that it's not always enough, right? It's true that in some cases, even if you use just UMAP or t-SNE, you can capture something about the spatial organization or temporal process, underline the data, but many times you can't, right? And then the question is, how do you use prior knowledge, and how do you constrain your reconstruction to also enforce what you already know about the data? And for example, with NovaSpark, what you're doing, besides the fact that it's probabilistic and that you can use other types of prior information, you're also saying that you know something about the structure of the tissue, and now you're trying to enforce your embedding to capture that, right? Like in the fly development example that I showed, right? So you don't know which cells go where, but you know that they should lie on, like in a particular way in space altogether. Or if you have some underlying symmetries in the system, and you're not necessarily looking for a 3-dimensional embedding, but maybe a 1-dimensional embedding with maybe a bifurcation or something like that, right? So in very easy cases where you can already see the full picture from just the straightforward embedding, then that's great. But in most scenarios, then it's not the case. And then you need to do something else and use your other attached prior knowledge to enhance the embedding.
Actually, I had another question. So how far you think you can go just from single cell sequence data to infer the temporal structure, temporal information of the system?
So what do you mean, how far can we go?
Like how accurate you could pin down the actual differentiation, like temporal differentiation process of the system, something like that.
Right. So that really depends on the system, right? It's hard to give like a one, you know, a number for this type of question. Yeah. But I will say that we're working on, you know, we're thinking both in cases. So we have several projects related to temporal reconstruction, both about how, you know, in cases where we don't have, where maybe the process is simple enough so that we can rely on just de novo reconstruction or a few marker genes, like in the case of maybe the cell cycle, or in cases where additional information would really help, like when we have lineage tracing information, for example, right? So how can we now get a useful embedding of the data when we have this additional layer of information that tells us something about how subsets of cells are transitioning, right? And again, thinking about it from a probabilistic point of view, and with these, but yeah, so I guess it depends, right? And as the system becomes more complex and more challenging, we would need more, more, you know, orthogonal data to complement the original single cell data.
Rudy, why don't you go ahead?
Hey, Nitzan. Thank you for the great talk. It was very illuminating.
Thanks.
I have a question about like how many layers you want you could disentangle in your data. You talk about spatial temporal information that you have. So you can add many more layers. And if you have to how do that?
Yeah, so of course, you know, ambitiously and optimally, we'd like to decouple all different meaningful layers of information in the data. But because it's challenging problem, then we're starting by thinking mostly about 2 or maximum 3 different layers. So the examples that I showed in this talk were all, you know, maximum 2 different layers, right? Spatial and temporal. But of course, you have many others, right? So you have, you know, we can also ask, can we now filter out these to expose different cell types, or, you know, maybe differently ordered along the process, or different interaction networks between different genes, between different cells. So I think in the slightly longer term, we'd like to be able to answer this question more generally for multiple different layers of information, not just 2. But yeah, but we're now starting from, you know, we're going step by step.
But of course, but for example, if you have more data, like I don't know, I'm just thinking in some resources, or so you have maybe another part of the information you can disentangle this part too, like in trying to put it in your analysis, like a probabilistic analysis of those.
Yeah. So conceptually it should work in that case. You know, and in very simple scenarios, we've done 3 layers as well, but this you know, this more general challenge is still work in progress. But conceptually it should be possible, right? It's not constrained to 2 different layers.
Thank you.
Okay. I mean, we probably should switch over, but I kind of have one more thing I'm thinking about, which is that last slide.
Sure.
We put up that maybe we can learn regulatory networks.
Yeah.
Here, and I'm wondering how you see those. Yeah.
What do I mean by that?
What do you mean?
Right. So
it kind of gets at this question of like, how many layers could you actually get? And we're saying like a handful versus, I don't know,
right.
these networks are like tens, hundreds of genes.
Right. So first of all, I'll say that when I say it, it's an ill-defined term, right? Saying layers of information, right? That can mean different things. But when I talk about it and thinking about communication networks, and I'm thinking about that as like a layer of information, right? We're trying to expose some coherent structure in the data. So that can be either spatial structure or temporal structure or like interaction structure in the data. So I'll say that at first. And then more concretely, there are these really interesting questions that we can continue talking about in a few minutes about how all these questions and approaches have to do with mechanisms and dynamics, right? So directly to answer your question, say we have single cell data, we recover an underlying temporal process like the cell cycle, right? So now we know how the different cells are, the different cell states are progressing along the cell cycle, right? So that's good. But then a very interesting question is to ask, okay, but what is the mechanism that leads to the specific structure of the cells in gene expression space, right? What is the and we can think about it from many different directions, but one of them would be if we assume that all the information is captured by the RNA expression levels of the cells, which of course is an approximation. It's not entirely true, but it may be a good enough approximation for us at this stage. Then what is the minimal set of equations relating these different genes and how, covering their interactions that would give us a dynamical trajectory that is similar to what we see in real data, right? And of course, it's very challenging because we have tens of thousands of genes and there are different ways to deal with that. But that's the core of the question, right? So can we now link these manifolds to models or systems of equations that actually describe the trajectories that we see in space?
And I guess part of that is like when you isolate the variability, let's say the circadian like angle, whatever, when you find the parts of the cells that correlate in the circadian realm, then you can maybe have more constraints on fitting.
Right, of course. So now, so what I talked about has to do with, say that the cells only encode a temporal process, then great. Once you have this representation, then you're good to go with now trying to understand how can you link that to mechanism. And I have some previous works about, how can you actually infer gene regulatory networks from these temporal trajectories, right? But of course, as you mentioned, in real data, as we talked about, right, there are these different layers, and some of them have to do with the temporal trajectory that you care about, and some of them have to do with other things. Yeah. So what you'd like to do, right, is filter out all these other things unrelated to temporal trajectory that you're trying to get the mechanism for, and then get hopefully a better representation for the mechanism using that approach.
Totally. Okay. Very cool. Thank you. Okay, if there are no more questions on the content, I can turn off the live stream.